Hello biology students, and welcome to the second video in the meiosis unit where we're going to be talking about the actual steps of meiosis. So meiosis, at first glance, like in this picture here, looks really similar to mitosis. However, there are a couple of key differences that allows us to make haploid cells at the end. So remember, the purpose of meiosis is to make cells that have half the number of chromosomes as normal, aka haploid cells, so that way when these cells, egg and sperm cells, combine, we get a cell that is a normal diploid. So first, let's talk about meiosis 1. Now, meiosis has two main divisions, meiosis 1 and meiosis 2. In the first division, meiosis 1, the first thing that's going to happen is we're going to get interphase. So interphase is not actually part of meiosis. It's, interphase, just like interphase of mitosis. So interphase happens first, before meiosis 1. And then meiosis 1 is going to have 4 steps. It's going to have what we call prophase 1, metaphase 1, anaphase 1, and telophase 1. And then these are going to be followed by cytokinesis, just like in mitosis. A key thing to remember during meiosis 1 is that we are separating homologous chromosomes. So in each homologous pair, one chromosome from each pair is going to one daughter cell, and the other chromosome in the homologous pair is going to the other daughter cell. So this is not like in mitosis. Remember, in mitosis, we were separating sister chromatids, but here we're separating homologs. And at the end of meiosis 1, we'll have 2 haploid cells that have replicated chromosomes. So here's a little overview picture. So you can see in meiosis 1, I'm starting with a diploid cell. In this case, we have 4 chromosomes. And then you can see during metaphase 1, the homologous pairs now are going to line up on the metaphase plate, and I'm going to take one chromosome from each homologous pair and pull them to either side of the cell during anaphase 1. Then during telophase 1, you'll see at the end, I get 2 cells that are now haploid, and then these cells have replicated chromosomes. So that's kind of a summary of meiosis 1. We're going to look at meiosis 2, which is this part of the picture later on in the video. So now let's talk about these steps in a little bit more detail. So in prophase 1, all the normal things that happen in prophase of mitosis are also going to happen in prophase 1. So the chromatin is going to condense into chromosome form, the nuclear envelope will break down, the spindle fibers will start to be built. However, something new is going to happen. During synapsis, which is part of prophase 1, we're going to get my homologs. Homologs is kind of shorthand for homologous chromosomes. So the homologous chromosomes are going to pair up. So the 2 chromosome 1s are going to pair up, the 2 chromosome 2s, all the way up to chromosome pair 23. We also get this thing called crossing over is going to occur in prophase. We'll talk about crossing over later in the unit. But just for now, note that it happens during prophase. Next, during metaphase, my homologous pairs are going to line up on the metaphase plate. So as you can see in this picture over here, the chromosomes now are lining up 2 by 2 and not single file like they did during mitosis. Therefore, because they're lined up this way, the spindle fibers attach only to the outside of each centromere. So I have one spindle fiber attached to this side and then one to this side of the other chromosome. And which side they attach to is random. So this spindle fiber could attach to this chromosome over here, this one could attach to this chromosome over here, or vice versa. The chromosomes then kind of line up randomly 2 by 2. This random lining up is called independent assortment, so just kind of note that vocab word in your head now, and we'll talk more about what independent assortment is later in a different video. And again, they are lined up 2 by 2, not single file. So next, just like in anaphase of mitosis, the spindle fibers are going to shorten and pull the DNA to either side of the cell. But in this case, if you look, we're pulling apart our homologous pair. So homologs are being pulled apart, not sister chromatids. So this is different than in mitosis. And then finally, in telophase 1 and cytokinesis, we're going to get the same things that we got in telophase and cytokinesis of mitosis. So the chromosomes will reach the poles, the cell will sort of pinch with a cleavage furrow. The nuclear membrane might reappear, or it may not, depending on how quickly we move into meiosis 2. And the key is now, at the end of meiosis 1, we have haploid cells. We have cells that only have one copy of each homologous pair, right? In human cells, we would have 23 chromosomes at this point in each nucleus. Okay, here's a picture. You can see in this case, in telophase 1, the chromosome decondensed into its chromosome form. You can see the nuclear envelopes are starting to reform, and then we're going to get cytokinesis next, where each of these 2 cells pinches into 2. So to kind of, again, to summarize, in meiosis 1, the big difference between this and mitosis is that our homologs paired up during prophase. Those pairs lined up on the metaphase plate during metaphase, and then the homologs were separated during anaphase, meaning that we have haploid cells at the end of meiosis 1. So now for the second half of meiosis, meiosis 2, now here is where we're going to separate sister chromatids. So at this point, we're starting with the 2 cells that we ended with at the end of meiosis 1. Remember, those cells were haploid with replicated chromosomes. Meiosis 2 has 4 phases, just like meiosis 1. We have prophase 2, metaphase 2, anaphase 2, telophase 2, and we're going to have another round of cytokinesis. And this, meiosis 2, looks just like mitosis. So we're going to have sister chromatids be separated from one another now. Okay, and when we end meiosis 2, we will end up with 4 cells. Those cells will be haploid, and they will have non-replicated chromosomes. So prophase 2 is going to look very similar, actually, in fact, identical to prophase of mitosis. So we're going to have the nuclear membrane breakdown. If it reformed during telophase, we'll have the centrioles migrate to either side of the cell, and the spindle fibers will start to form. Also during prophase, the DNA will condense into chromosomes. In metaphase 2, the chromosomes will now line up single file down the center of the cell, okay, and the spindle fibers will attach to both sides of the centromere. This looks exactly the same as metaphase of mitosis. In anaphase 2, the spindle fibers will pull the sister chromatids apart, right? So that looks just the same as mitosis. So again, now we're separating chromatids, just like mitosis. And then in telophase 2, this will look just like telophase of mitosis, right? My nuclear envelopes will reform, DNA will decondense, and we'll get cytokinesis. The 2 cells will split. So if you think about, at the very end, how many cells are made in telophase for one that started in meiosis? We get 4 cells total. We started with 1 cell, that became 2 cells during meiosis 1, and then those 2 cells became 4 cells during meiosis 2. So if in prophase 1 the cell had 10 replicated chromosomes, how many chromosomes are present now? Well, we're going to have half the amount of DNA because now we have haploid cells. So we'll have 5 unreplicated chromosomes. We started with 10 replicated chromosomes. After meiosis 1, we had 5 replicated chromosomes, and now we have 5 unreplicated chromosomes in each cell. So then finally, these 4 cells that are produced, these 4 cells will develop into egg cells if we're in a female, or sperm cells if we're in a male. Now there is an exception in a female that we'll talk about later, but for now, just know that those 4 cells become either 4 sperm or 4 egg or egg-like cells. And remember, egg and sperm, we call the gametes. So finally, to summarize, we start with interphase, where we replicate our DNA. We then get meiosis 1, where we separate homologous pairs. And then finally, we get meiosis 2, where we separate sister chromatids. So at the end, our 4 products, either egg or sperm, have exactly half the amount of DNA than our original cell did. So we have succeeded in our goal, which was to create cells with half the amount of DNA.